This experiment details how to obtain diffraction quality crystals of a protein bound to DNA containing a tandem hemimethylated GATC repeat. To favour crystal packing of the protein-DNA complex, start with a rational design of the DNA length, the GATC spacing, and the DNA ends. Proceed with the complementary single-stranded oligonucleotides to purify the DNA, and then anneal to form the hemimethylated DNA duplex. Then, mix in the purified SeqA to form the complex. Next, find optimal conditions to grow diffraction quality crystals of the protein DNA complex with crystallization trials using commercial sparse matrix screens. Demonstrated results show the effect of varying DNA length, GATC spacing and ends on the diffraction quality of the SeqA-DNA crystals.
The goal of this work is to obtain diffraction quality crystals of SeqA bound to DNA, but it can also help to understand the general variables that need to be considered when designing DNA duplexes for crystallization of other protein DNA complexes. Demonstrating this procedure will be Seon Chung, a postdoctoral fellow from my lab.
This study uses a variant of the DNA replication regulatory protein SeqA that forms stable dimers and has a shortened linker region between its oligomerization and DNA binding domains. This variant restricts DNA binding to tandem GATC repeats separated exclusively by one turn on the DNA. First, transform BL21DE3 cells with the plasmid encoding dimeric SeqA under the control of the T7 promoter.